That's what's going on. Thanks, Brandon. Or who was it who said I was muted? Now it makes much more sense. Oh, Kosar, thank you. I was wondering what was going on. I was threatening to give the entire lecture in Spanish. Nobody said anything. All right, we ready? Is Stoke high? Is everybody ready for the next exciting continuation of this? Yeah? Yes? Yes? Stoke is high. All right, well, let's do it. So catabolism of amino acids. Last week, yes, let's do it. Last week we learned about anabolism of amino acids, and this week we'll learn about catabolism, which means we're going to figure out how we made those wonderful, amazing molecules, and we broke them down into little bits, and how we get those things back into our system and use them. Also, we're going to figure out how you transform a Thanksgiving turkey into, energy to run and jump and dance and sing. So woohoo, let's figure out, how are you processing your turkey? Oh wait, I'm trying to advance the slides on the wrong computer. Here we go. So as you recall from our lecture a couple of days ago, anaplerosis is the process by which we add carbon to the citric acid cycle in a net way. So when we add carbon in 2 carbon units, are we adding net carbon to the TCA cycle? That's a question, chat. Yes or no? Are we adding carbon to the TCA cycle in a net way when we add them in? No, no, no. All your no's are correct. And why is that? Why, chat? Why can't we feed our TCA cycle like and make the, yes, because we are losing them all as carbon dioxide, right? So this is a really fundamental and a critical principle to get your head wrapped around as mammals and, vertebrates, like many vertebrates. I don't actually know how far evolutionarily this extends. Think about that. Think vertebrates, chordates. I don't know how far this goes, but so microbes and some plants have a way around that, as you guys remember. But for all of us who stand upright and have emotions, though we don't have that ability. And so as a result, we have all these different ways of feeding our TCA cycle, and most of that comes from amino acids, and that comes from amino acid catabolism. So we've already learned a fair amount about glutamine catabolism and how that gets into the TCA cycle. In fact, you've done that in your tutorials now, where you've done isotope labeling work, where you've added in glutamine and seen it appear in alpha-ketoglutarate and some other things. But there are quite a number of other ways you can get carbon in. You can get it in from aspartate, you can get it in from branched chain amino acids, and a variety of other things. So, so let's move on. So this should look pretty familiar. Now, we talked about before what the source of carbon was for making amino acids. And we said there were some general categories. We had some that originated from glycolysis. We had some that originated from the TCA cycle, et cetera. So we had a few different places. Well, not surprisingly, when we break the amino acids down, they also go back into central carbon metabolism, and they, by and large, go to the same places as the biosynthetic pathway routes, although not always. There are some, there are a few exceptions here we'll look at. So while humans don't have the ability to make many amino acids, we do have the ability to break down all of them, right? And that makes some sense because they're coming in our diet. If we can't break them down, that's an issue, right? So here we're going to look at the various ways we break down amino acids. And there are 2 major categories. One is there are glucogenic amino acids, and that means amino acids that ultimately can add net carbon to the TCA cycle, or can be broken down into 3 carbon units or more, right? Is the net product. Those are the glucogenic amino acids. And then there are the amino acids that ultimately get made into 2 carbon units, or things that can't be catabolized into 3 or 4. And you'll understand that distinction here in a little bit. But like, so the kenogenic amino acids include the leucine, isoleucine, tryptophan, threonine, lysine, leucine, phenylalanine, tyrosine, these kinds of things. So they're the sort of the aromatic things, and branched amino acids kind of fall into that. And by and large, the the other amino acids are glucogen. Okay, so let's talk about these now as a group by group. So first, let's talk about all the amino acids that can be broken down into pyruvate. I guess we already mentioned how alanine, and I think that's on this last slide here, we already mentioned how alanine gets into pyruvate. And that was in our last lecture as via alanine aminotransferase. Now let's talk about how some of the other ones get into pyruvate. One is serine. Serine can be deaminated. And this should be no surprise to you now in seeing this recurring pattern that the cofactor for this deamination is PLP, pyridoxal phosphate. And also, not surprisingly, if you remember our last lecture, glycine is also can be converted into serine, and serine can be converted into pyruvate. And what also shouldn't be a surprise is in order to convert glycine into serine, it involves a methyl transfer. That's just the reverse of the reaction we learned before, and tetrahydrofolate is involved as your cofactor. So this is going to be a recurring theme, and what you see is how biochemistry works. When you need to do a 1-methyl transfer, you call on tetrahydrofolate, and when you need to do a deamination or amination kind of reaction, you'll use pyridoxal phosphate. So this is just how cofactors work, is they enable specific kinds of chemistry, and you see them as repeating themes. This is how you get to pyruvate from serine and glycine. And then threonine is also catabolized into pyruvate, and it does that in a couple of steps. First, you have it. Threonine is oxidized into 2-amino-3 ketobutyric acid. So this is butyric acid with an amine on it, and then you lose a acyl-CoA off of it, 2-carbon unit, and you end up with glycine. Well, that glycine then can then enter into pyruvate just the same way you saw in the last one. Make sense? So it makes an acetyl-CoA and a pyruvate in the case of threonine. In the case of cysteine, the cysteine is oxidized into cysteinosulfonate. And then ultimately, again, pyridoxal phosphate is used to get rid of that amine off of the side chain there, and that after another step of desulfurylation ends up into pyruvate. Make sense? For each of these pathways, I'm going to expect you to know the precursor, the product, and any major cofactors involved in the chemistry. Okay, so for each of these, and we'll see is that when they get beyond 1 or 2 steps here, like they can start to get kind of hairy. So what you should definitely know what they come in as and how they go out, and any major cofactors involved in the transformations. Tryptophan catabolism. So this is, of course, the famous molecule out of Turkey. We'll talk a little bit about dopamine and neurotransmitters, or where people always blame the laziness that they feel after Thanksgiving on tryptophan and tryptophan catabolism. It may have something to do with the bottle of wine that people drink at Thanksgiving, but nobody counts that for the down feeling they have after eating turkey. But so here is how tryptophan is broken down. So first off, it's got this monooxygenase step here where tryptophan is oxidized in this very first step to make this funky molecule N-formyl kynurenine, and that is then broken down into kynurenine. So kynurenine is actually an amino acid we see frequently in our metabolomics assays here in the group, and it's something that's a catabolite, very common catabolite that we see of microbes. So we actually keep an eye on kidney. It's a friend of ours, one of our molecular friends. But that molecule can then be oxidized again by a monooxygenase into this 3-hydroxy kidney molecule, and ultimately an alanine is released from that, and then you get this really funky a molecule called 3-hydroxyanthralenate, and that can be further catabolized in a series of steps, ultimately into acetoacetate and acetyl-CoA. So this anthralenate is another molecule that we see routinely in the mass spec, and is a catabolite of microbes. Frequently when they're eating tryptophan, we'll see the anthralenate or the kidney as metabolic catabolite. So not everything necessarily gets broken all the way down into its component parts, particularly in microbes that are trying to grow fast. Sometimes they'll secrete the carbon out. But if they are broken down all the way to central carbon metabolism, this one comes out as acetyl-CoA. Yeah. So now let's talk about the amino acids that go in as alpha-ketoglutarate. Many of these we've already seen before, glutamine, glutamate, of course. But let's just take a look at those pathways again quickly to remind ourselves. So glutamine can be converted into glutamate here via the glutaminase reaction. So that process loses a nitrogen or an ammonium. And once you have that glutamate, obviously that can get back to alpha-ketoglutarate via a variety of reactions that we've talked about. One is glutamate dehydrogenase, which is ultimately produced as an ammonium and an NADH, or another version of that is aspartate aminotransferase or alanine aminotransferase, or any one of the other aminotransferases that we've talked about in the biosynthetic reactions or the anabolic reactions. But ultimately, those things all make alpha-ketoglutarate. Another really important class of reactions here, of course, is proline. You'll be very pleased to see the same pathway in reverse of the one we learned before. So proline gets turned into this prolyl 5-carboxylate and this glutamate gamma-saminoaldehyde glutamate, ultimately alpha-ketoglutarate. And similar pathway for arginine. We're going to be learning more about it in the urea cycle, but this a very important enzyme called arginase ultimately takes the arginine, takes that funky, really funky side chain off of it and makes an ornithine, which we'll be learning a lot about here in a minute. Arginases, as it turns out, in nitric oxide synthase are really important enzymes, not only in amino acid metabolism, but also in immune function. You'll find a lot of literature about microbes or pathogens that use arginase as a tool to help evade immune system or help to mitigate the response of the immune system. And ultimately, this is because nitric oxide synthase, one of the tools your immune system has to combat microbes, uses arginine as a precursor, and so arginase can break that down and sort of be used to combat your immune system. So you should pay attention to this. It's a cool enzyme. It's important for you. Other important ones, histidine also ends up in alpha-ketoglutarate, and it goes through this series of catabolic steps. Uraconate here, this first step after the deamination. Again, this is something that we see that microbes produce a lot. Uraconate is one of the things that we look for when we're trying to diagnose bloodstream infections for certain microbes. Uraconate is made by those pathogens. So these things aren't random whatevers. Some of these things might end up saving your life, and we'll talk about in a later lecture how we're harnessing this amino acid catabolism and other central carbon metabolism to make rapid diagnostic tools for infectious diseases. But they're based on these kinds of molecules, detecting them and understanding them. So similarly here, we have a number of catalytic steps. I expect you to know the precursor and the final product and any cofactors along the way. So we see that this one has a tetrahydrofolate. So what do you guys guess? What's what kind of chemistry is going to be going on here? We're going to have a 1-carbon transfer going on at some point, right? Tetrahydrofolate. So then some other amino acid pathways along the way are asparagine aspartate. They're made into oxaloacetate. We already talked about these in our last lecture, but to remind you, oxaloacetate is converted into or I'm sorry, asparagine is converted into aspartate here via this, well, this in this case is a deamination reaction, making an ammonia, and the aspartate is converted back into oxaloacetate using the same enzyme we learned before, the aspartate aminotransferase. And of course, that amino transfer reaction is going to involve a pyridoxal phosphate, as they all do. Now, on to some of these things that are feeding into succinyl-CoA. I have some amusing history here with these reactions we could talk about sometime if we have time, but isoleucine, methionine, valine, threonine, valine all ended up being broken down into succinyl-CoA. And these reactions involve some somewhat scary looking reaction mechanisms here. This is one that goes through, we talked about before, and methionine, it goes through this S-adenosyl methionine reaction. SAM is a very important methyl donor. It serves as a methyl donor for other reactions. So it serves a number of functions beyond just methionine. Anyway, so it goes through this series of steps here, ultimately making, it has cysteine coming off and alpha ketobutyrate, propionyl-CoA, ultimately making succinyl-CoA. Okay. Again, you're expected to know the beginning point on the pathway, the end point on the pathway, and any major cofactors along the route. Now on to our acetogenic amino acids. We have our leucine, isoleucine, tryptophan, etc. So your phenylalanine and tyrosine are broken down by a series of steps here. So phenylalanine is oxidized into tyrosine is the first step. We talked about this step last time. We'll be covering more about this in the phenylketonuria and the enzyme deficiency that can result in phenylalanine problems because of the loss of that enzyme. Then it goes through a number of other catabolic steps that ultimately bring it to fumarate. And that fumarate is then catabolized into acetoacetate, right? And ultimately acetyl-CoA. So the key points here is that it comes in as phenylalanine or tyrosine, and that it has these acetyl-CoA as the endpoint. Okay. So lysine catabolism. Lysine catabolism also has several steps here, but ultimately you have a alpha ketoglutarate serving as a cofactor, not a cofactor, but part of the carbon chain here, and you make this crazy molecule, saccharopine, and that molecule ultimately, when you make it, has a glutamate that comes off as a product. Is a very complicated way of getting rid of a nitrogen, and then it goes through this amino adipate semialdehyde and the intermediate, which is actually a precursor for other pathways. And then it can also be catabolized into this compound called alpha amino adipate. And that adipic acid, alpha amino adipate, they come up in biochemistry pretty frequently. We see them as they're a non-traditional or non-standard amino acid, and we do see them being in microbial systems and serving a variety of other biological functions. But ultimately, you have the endpoint of this pathway is alpha ketoglutarate, just like acetyl-CoA. And again, when you're expected to know the beginning points of this pathway, the endpoints of this pathway, and any major cofactors along the route. Okay, branch chain amino acids here. We're getting near the end. So leucine, valine, isoleucine, all these guys have this peculiar side chain structure here that makes them a little hard to break down. And so what we do is first you got to get rid of the nitrogen. That's done with this pyridoxal phosphate, and that pyridoxal phosphate is, the same kind of chemistry that we talked in the other ones. This transamination reaction. We mentioned this in our last class, and then from there it has to get broken down further, and it goes through this branch chain keto acid dehydrogenase complex. So that complex actually has a almost identical reaction mechanism and series of reactions as the pyruvate dehydrogenase complex. So it's a very similar enzyme complex. Came as a result of probably as a result of enzyme duplication. So it just has a very subtly different substrate specificity, where it takes these alpha keto acids like alpha ketocaproate and it converts those ultimately into acetyl-CoA or these acyl-CoAs, sorry, after this oxidation event. And that ultimately produces a variety of precursors here, branch chain precursors with double bonds in them that ultimately can be broken down into either acetyl-CoA, succinyl-CoA, or propionyl-CoA. And but the important things to remember here are where the starting points are, the endpoints, and the fact that it goes through this branching amino acid dehydrogenase complex, which is very similar to pyruvate dehydrogenase. Okay, so we've blasted through quite a number of pathways here, talked about how amino acids get broken down. And I think you should have an appreciation now that your body has a lot of pathways. So like up until this point, we've been spoon feeding you pathways like one a week. But now it's time to open your eyes to see that your body has thousands of reactions that it does. And ultimately, they all are ways that bring the things that we eat or the things that we need to connect those carbon molecules with things in central carbon metabolism. Amino acids are a really great example of that. And we've evolved really clever ways to get all the molecules that we need to make our bodies into basically all entering into glycolysis, the TCA cycle, pentose phosphate pathway, which is why you guys have been taught these pathways in so many of your courses, is they're really core to our survival. Now, after you've broken down these pathways, we've left out one important detail, which is that humans are in a weird state. Well, heterotrophic organisms that eat other organisms, are weird in that we are in a constant state of having too much nitrogen around. We got to get rid of our nitrogen. Now, all the free living organisms of the world have to contend with getting nitrogen, getting access to it, because most of it's locked up in some not very useful forms like nitrogen gas or nitrate, forms that are hard to deal with. We have this wonderfully accessible organic nitrogen all the time. And so our issue is that we mostly have to get rid of it. And so let's talk a little bit about how you get rid of it. So the issue is that the free ammonium is toxic, right? So it destabilizes our, it's a nucleophilic. It destabilizes our biomolecules, and it has it's a nasty molecule to have a lot of it. So we have to get rid of it. Now, by we I mean big multicellular organisms. We have to get rid of it. Now, if you are a microbe living in the sea and you happen to be having, in a soup of lots of amino acids, or if you happen to be a crab or a fish or anything that has free access to water, the most easy way of getting rid of ammonia is to secrete it directly. And that's the strategy that a lot of aquatic organisms use, or microbes when they're grown in rich cultures. They just secrete they can frequently just secrete the ammonium directly. That's because it's soluble. It comes right out in the gills in fish. So other organisms have different strategies for dealing with it. Obviously, we're not living in a bath of water all the time. So we get rid of it as urea. So the reason we do this is we detoxify it into a form that's less reactive, this urea molecule, and then we secrete that out in our urine. Now, the problem with our strategy is that urea has to be dissolved in water. And so we have to pee out a bunch of water every time we want to get rid of urea. And so if you're an organism that's dealing with, drought or wants to be more water efficient, then there's actually a better route of getting rid of it, and that's to get rid of your water as uric acid. And so that's what birds and many insects do. This is the white part of bird poop, is that's how they get rid of their ammonium. And the neat thing about that is it doesn't require anywhere near as much water to get rid of, because it's, they can secrete it as a semi-solid. I think we just talked about this already. Why is ammonia toxic? Those free electrons give it a problem. Hey, I advanced my slide, but it did not advance there. What's going on there? Okay, here we go. So how is ammonium produced? So it comes from a lot of places. We've already talked about a whole bunch of pathways that make ammonium through their amino acid catabolism. But here are a couple of examples as reminders. It can come directly from serine, or it can come directly from threonine, for example, or a variety of other amino acid catabolism steps. Well, it can produce nitrogen directly or ammonium directly via these deamination reactions. And after you've oh, right here, and of course, naturally, we've already talked about this deamination reaction here of glutamate. Here's another example of that. Quite a few of the ones that we just talked about in the last series of slides ultimately take an amino acid and ultimately make a alpha keto acid and ammonium. So something that's peculiar about this and people is like, by and large, most of these reactions, are occurring in liver. But something that is funny is that branched chain amino acids are actually in people are catabolized in the liver, or in the muscles, not in the liver. And so with this branched chain amino acid catabolism that we were just talking about, like this ultimately produces ammonium in the liver that's used in alanine, and through this branched chain amino acid transaminase reaction that we learned about, and that makes alanine, which is then transported to the liver, where it's ultimately broken down into ammonium and enters the urea cycle. And we'll talk about that in a minute. So in humans then, and in other like upright vertebrate things, or many upright vertebrate things, mammals, I should say, once we've made broken down all these amino acids and made our ammonium, we have a series of reactions involving multiple compartments intercellularly that ultimately coordinate to get rid of this molecule as urea. So this is the final end product, the thing that we pee out inside our kidneys. And this is the starting point up here, this ammonium that comes from all these different amino acid catabolic pathways that we just talked about. So naturally, many of these things, like we're particularly highlighting glutaminase here and glutamate dehydrogenase, because glutamine is the most abundant amino acid in our body, and so this is a very major route for nitrogen catabolism and excretion here. But here are a variety of ways where we get this ammonium. So the very first step in the urea cycle actually starts with this ammonium, where you have to get it back into a form that's reactable. So the very first step is a reaction we call a carbamyl phosphate synthetase 1, or CPT1. And we'll see that this is also a reaction that's used in nucleotide biosynthesis, or a very similar enzyme. But so CPT1 is the one that's used in the urea cycle. And what it does, it takes this reactive nitrogen here and it puts a phosphate on it. And this allows you to do some chemistry with it. The ammonium itself doesn't really want to, it's difficult to cajole into the right kind of biochemistry reactions without activating it. So we activate it in this way. Okay, this process takes an ATP, or it takes 2 ATP and makes 2 ADP. In effect, we'll see each of those reactions here. That's the net product of that. in order to get this carbonyl phosphate. So this is the starting point. This is the activated ammonium. And then it involves this series of reactions that we're going to learn about step by step here, where we make citrulline, and the citrulline ultimately is converted into this arginosuccinate. The arginosuccinate is ultimately becomes arginine with fumarate coming off and ornithine, right? And the final product here is urea, and this thing ultimately goes out and is peed out by the liver. So we'll talk about this cycle. So the very first step here is carbonyl phosphate synthetase 1. We know it's since it's got the word synthetase, we know that it involves hydrolysis of ATP. And so there are two CPTs. One of these is CPT1 is in the mitochondrial matrix, and this is the one that we use for the urea cycle. And then the other one that we're going to learn about later is the one I was just mentioning, is this involved in pyrimidine biosynthesis. Both of them involve taking nitrogen, inorganic nitrogen, and a bicarbonate, which the dissolved CO2 is in the bicarbonate form, and then combining them to make this activated carbonyl phosphate molecule that you can do chemistry with. So this reaction ultimately produces this carbonyl phosphate that I was talking about, and that carbonyl phosphate is the is the activated ammonium bicarbonate, a molecule that you can use to do chemistry. So the the actual biochemistry looks something like this. You start with the bicarbonate. You end up with this carboxyphosphate intermediate that you undergo, and then you have this nucleophilic addition, this additional elimination reaction that gets rid of the nitrogen and substitutes it for an amine, and then for this nitrogen group, and then that is ultimately phosphorylated again to make your carbonyl phosphate, and you have your activated molecule that you can use for doing biosynthetic reactions. So after you've made this molecule, if you're going into the urea cycle, the next step is you take this carbonyl phosphate and you add it onto this molecule ornithine, which we'll see is the product that's recycled through the urea cycle. So ornithine is structurally very similar to arginine, as you guys probably recall from a couple of slides ago when we talked about the arginine catabolism. And so via this ornithine transcarbamylase enzyme, you end up transferring that side chain onto this molecule, the side chain carbonyl phosphate moiety there onto this citrulline, which is a funky, cool 1, 2, 3, 4, 5 carbon molecule here, and ultimately that releases an inorganic phosphate. Then the next step is you take arginosuccinyl synthetase, takes ATP in that citrulline molecule that we just made, and ultimately makes a pyrophosphate, so it's 2 ATP equivalent, and it makes a citrulline AMP intermediate. And a spartate comes in, adds to that, and it makes an arginyl succinate as an end product with an AMP as a leaving group. So the net reaction is you have a citrulline, an ATP, and a spartate making arginosuccinate, 2 pyrophosphate and AMP. So this is a 2 ATP equivalent reaction. After you've made this wild arginosuccinate molecule, the next step is arginosuccinase. So it breaks this down into arginine and fumarate as component parts. Obviously, fumarate can go right back into the TCA cycle, and arginine is an amino acid that and we learned about, and it's now just one step away here from your urea. You should start to recognize this structure here as looking very similar to the urea. And then this arginase, this enzyme I was telling you about, will ultimately take that arginine, clip it off into ornithine, the molecule we started the cycle off with, and our urea. And that urea end product ends up going to our kidney. Now, we get rid of huge amounts of urea over our lives, and as you're showing here, 10 kilograms of urea each year we get rid of. Which we solubilize in our pee, and this is one of the main limiting factors when we talked about our rabbit starvation, and the protein starvation, or when people are starving to death by eating only protein meals. The reason is why ultimately it takes a lot of capacity here to get rid of this much of nitrogen, and so humans are running just near our capacity for being able to process nitrogen. Okay, so this entire process is pretty energetically expensive. It takes ultimately 3 ATP, aspartate, and ammonium and CO2, and it converts that into urea, 2 ADP, two phosphates to an AMP and a pyrophosphate. So it's a lot of energy goes into making these things, and a fumarate. It comes out. So yeah, this is not a free process. It's something that requires some energy to get rid of our nitrogen. One of the important points about how we turn on and off this process, we do it through the very starting point of the reaction. So the CPS1 is allosterically activated by a molecule called N-acetylglutamate, or NAG, this is NAG is a molecule that's made when there's a lot of glutamate around. And it can also be high when you have you're fasting or you have high protein diets. And it's made just from acetyl-CoA and glutamate. It makes this acylated amino acid. And this is one of the allosteric activators. As a sidebar, these acylated amino acids here, these can be very abundant in metabolite extracts. We see them all the time when we do metabolomics, and it's not just acylated glutamate. We see acylated all kinds of amino acids, acylated serine, acylated all kinds of things, and some of them, most of them don't have known functions. So if any of you are interested in solving a biochemistry riddle, tell me why there are millimolar concentrations of acylated amino acids in most cell extracts. And that's a topic for another time. Okay, so in summary, we've covered a lot of ground today. What are the things that you need to know? So you need to know how we take amino acids that we eat and convert them into molecules that you're familiar with, things from the central carbon metabolism. You need to know how you get from the beginning point into the end point, and the most important cofactors involved along the way. You need to be able to explain how we get rid of that ammonium after we've produced those molecules, and how that goes through the urea cycle. And then we need to explain why different organisms use different strategies for getting rid of ammonium. So that's a summary of the materials we have today that are expected, and happy to have questions if there are any. I'm sensing a high degree of stoke from the chat. Thank you, Tarek. How does NAG regulate CPS1? Good question, Jeremiah. This is just an allosteric regulator. CPS1's turns on and off the pathway, and so NAG just acts as an allosteric activator or suppressor. Overwhelming stoke from Haley. That's great. Glad to hear that. You know, it's hard not to be stoked when you find out how you turn your turkey into central carbon intermediates. Pretty awesome. I think in our next lecture, we will cover what happens when these pathways get messed up. And we'll talk about how energy drinks and other things relate to these pathways. And we'll talk about how your body hijack some of these pathways to make other molecules that are useful, like secondary metabolites, like neurotransmitters and this sort of thing. Yeah. Any other questions? Looks like the chat is silent when I'm done.